But unfortunately, if you have a compartment that's free, or one of these high health farms, and if your biosecurity isn't perfect or you economize on anything and you cut corners, you get it. So we've had several of them break. And one of them that I looked at recently, they were using a disinfectant that was totally ineffective against African swine fever. And it's actually quite a fragile virus. There are a lot of disinfectants that work for it, but that particular one didn't. And so there's little things like farmers must make sure that the label does say, yes, African swine fever, not just almost any pathogen we will kill. No, that doesn't work.
Well, good day. My name is Ruud Zijlstra, University of Alberta, and today I have with me Dr. Mary-Louise Penrith, who is located in Pretoria, South Africa. Welcome, Dr. Penrith.
Thank you very much. Thank you for having me.
And today's topic will be African swine fever. What do we know after decades of knowing about African swine fever? So as a first opening question, Dr. Penrith, I would have for you, how did you get interested in African swine fever?
Well, I got interested in African swine fever because I was working in the pathology department at the Ondersupport Veterinary Institute, which is our main, it is in fact still, it's a reference laboratory for African swine fever in Africa. I had completed my veterinary degree. I had a long career because I qualified as a zoologist first, and I only qualified as a vet when I was in my 40s. But that's still a very long time ago. So, and I fell in love with pathology because it gives you the answers. So I started in the institute, and not long after I'd been there, I got a bunch of pigs to do postmortems on. They just had an E. coli problem, but I apparently did quite a good report, and so the head of department decided I should more or less be the pig person. And of course, African swine fever has been in Africa. We've been known about it for more than 100 years. And our institute was very involved in the early research on African swine fever, not the very earliest, which was in Kenya, but in the 1920s already. So pigs and African swine fever went together. And when the head of our Exotic Diseases Institute, which is at that stage separate, was asked to do consulting by the Food and Agriculture Organization in Mozambique for an outbreak of African swine fever, he felt that he was really a foot and mouth disease expert as well as an African swine fever expert, but he preferred foot and mouth disease, and he thought somebody else should be trained to do the consulting on African swine fever. It was just after, it was 1995 when South Africans had become acceptable in the international world again. And so I went with him to Mozambique, and I had read up about African swine fever, and I just got absolutely fascinated by it. And then he was supposed to do a consultancy in Kenya the next year, which was what I was then going to take over. And somehow, not long after that, in 1996, African swine fever exploded in West Africa, where it wasn't endemic as it was in Eastern and Southern Africa with the warthogs. So I got involved in that, learned some French in the process, and somehow I just kept on being asked to go back to do more consulting as more countries got infected. And it's just been, and then what I really saw was how very difficult it is for the pig farmers, and particularly in those countries, it was very often the very small-scale farmers as well as the very small commercial farmers, because you don't really get any very big commercial farmers in Africa. And it was very much, the pigs were very personal to them. And there was, of course, a lot of culling, and I got a bit against that. So wanted to look more at African swine fever to see if there could be alternatives to massive culling. And so I sort of am known for that, I guess. And then I got involved in a resistant pigs project in research. And I've just stayed with it. And then, of course, when it exploded again in 2007 globally, well, I've stayed involved. I'm getting far too old to be paid by the FAO anymore, but I still work with them a little bit on training and so on.
Interesting. Yeah, thanks for nicely describing. Of course, the topic is not nice from a pig producer perspective.
Most teams don't have a strategy problem. They have a systems problem. Tasks scattered across tools, goals buried in spreadsheets, and metrics no one actually checks. Zentrix OS fixes that. It's a true business operating system that brings tasks, goals, and performance metrics into one connected workspace. So your team always knows what to do, why it matters, and how they're tracking. No more juggling apps. No more missed accountability. Just a clear, organized way to run your business the way it was meant to be run. If you're ready to stop managing chaos and start managing with clarity, Zentrix OS is built for you. Visit zentrixos.com and take control of how your team operates.
Thanks for describing how it kind of evolved in Africa. And as you already indicated, now in other parts of the world as well, we're dealing with African swine fever. So what fields of research have received the most attention since African swine fever went global?
Well, I would say, and probably even when it wasn't global and it was just an African problem, I would say vaccine development was absolutely top of the list.
Yep.
We still don't have a guaranteed safe vaccine. A lot of progress has obviously been made, but I can just say there that while in the 1960s they were doing research, 50s and 60s they were doing research on developing a vaccine for African swine fever in Angola, which was in an overseas department of Portugal, because Portugal had got it in 1957. And by the 1970s, they had a vaccine, a live one, which they had attenuated by many passages in cell culture. And they released 400,000 doses into the field. And unfortunately, at that point, they discovered that it wasn't safe. Instead of, it protected some of the pigs, it didn't protect some other pigs, but a lot of the pigs became chronically infected and remained chronically infected, which was very unpleasant. It was difficult to recognize. And unfortunately, I think in recent vaccine development, we seem to have made the same mistake with the Asian vaccines. They look really good, and the problem is you can test vaccines in a laboratory with SPF pigs, young pigs, and works perfectly. And then you start putting it into all the pigs in the field that are suffering from mange and malnutrition and pregnancies and all sorts of things, and may already have African swine fever, and you're going to have a very unfortunate situation, which is unfortunately happens in places.
Is there anything specific about the pathogen that makes vaccine development so difficult?
Yes, it's actually entirely to do with the pathogen. I think it's a very, very large DNA virus. It has a plethora of proteins. I can't remember offhand because I'm not a virologist, but it's got a very, very large number of proteins. Mm-hmm. And we don't know what all of them do at this stage. People are still working on them, finding new functions, finding out which ones are virulent, which ones. And in fact, it has very good ability to evade the immune system. So it attacks the macrophage monocyte system, but it also causes apoptosis in lymph. It doesn't multiply in lymphocytes, but it does cause damage.
Yep.
And then the antibodies, you do get antibodies produced, but there are other antibodies that neutralize them. And it's really a huge problem. So depending on humoral immunity, which is what they're still doing, is not entirely satisfactory because it works for homologous virus, but it doesn't necessarily cross-protect against heterologous virus. And there are 23 known genotypes. Well, 24 have been described, but one has been retired, but 23 known genotypes, and it doesn't cross-protect the virus. The vaccines that they've got are for the genotype 2 virus, which is the main one circulating in the world at the moment, but it doesn't cross-protect against the genotype 1 virus. And unfortunately, we now have a hybrid recombinant genotype 1 and 2 virus, which, well, a lot of us think it's probably started off as a laboratory construct.
Yeah.
Because some old vaccine, genotype 1 vaccines, which were in the first outbreak outside Africa, were imported into a country where there was the genotype 2 virus, and that's where it all happened. And was molecular, that's the one great thing with molecular genetics, you know exactly what you're dealing with in the way of the viruses. But that thing, that recombinant is very dangerous because it's very virulent and there's no vaccine to fix it. But very good news is that there is now, people are working on a therapeutic based on knocking out one of the, it seems to be a very important virulence gene in the virus, and that's looking very promising. How expensive it would be to use it, I'm not sure. But
Yep, interesting. What region has been most prolific in terms of research outputs to deal with African swine fever?
Asia, definitely Asia without any question. Ever since it hit China in 2018, there's been an exponential explosion of a lot of very good, very useful research, almost all on the virus, not too much on epidemiology or anything, but all on the virus and on vaccine development. And it's been tremendous what's been done there, because it's certainly, particularly in Vietnam, they've done some very good research, apart from developing the vaccines that are just not quite as safe as they should be. But they've also done very good work showing that there isn't a long-term carrier state in recovered pigs, which was very good news to all of us, because it does actually mean you don't have to cull out pigs that have
survived. Yeah.
For poor people, that's very important.
Yep, of course. Yep. Yeah, and of course, since Eastern Asia got affected by African swine fever, the local emergency dealing with the virus, of course, has gone up drastically as well.
Yes, and also it's been very strongly coming out and has been emphasized in a lot of publications from all over that it's really a very human-driven disease, because people are what keep pigs and what decide how they're going to be raised, where they're going to be sold, how they're going to be used, and the very simple but good biosecurity can definitely protect the pigs against African swine fever. So there's been a lot of input, and a lot of the American research has focused on risk assessment and on biosecurity.
Because people carry the virus from one herd to another.
You can do that on dirty boots, yes, and also, the main ways it's moved around, and that hasn't changed over the centuries. We have a different situation because we have the so-called sylvatic cycle in which the virus evolved in warthogs, which are very extraordinary-looking African pigs, wild pigs, not closely related to domestic pigs at all. But we think the virus involved in ticks that live with the warthogs, and the virus co-evolved with the warthogs, which don't get sick at all. But when the ticks happen to bite a domestic pig and they're infected, then you've got big problems. But it's not like that anywhere else. Everywhere else, it's a strictly pig people cycle, and pigs unfortunately include your wild boars, your Eurasian wild boars, because they are simply the ancestor of the domestic pig. So they have become a bit of a problem, because if you've got there isn't a carrier state, but if you have a large enough population of pigs that are in fairly close contact, you can have maintained circulation forever because it'll burn out in a very small population, but it won't burn out in a large population.
Well, let's perhaps explore that a little bit. So what have we learned about innate immunity to African swine fever? And I think you related that already to warthogs.
Some very interesting work was done in Edinburgh on warthog genome, and they found a particular gene that appeared to be associated with the resistance in the warthogs. I don't know how they did it. I'm very much a veterinarian and not a molecular person, but it was, and they managed to engineer that pigs had that, giving them that gene, putting that gene into their genome. Unfortunately, they were not protected. They took a little longer. They were slightly protected. They took a little longer to get sick, and some of them didn't get so sick, and some of them did recover. But they were not, it certainly wasn't something that you could use. And the other African common wild pig is the bush pig. There are two species, the red river hog and the bush pig, but actually genetically they're probably subspecies. And they're also completely resistant, although they don't form part of the epidemiological cycle because they don't live in burrows like the warthogs do, so they don't have ticks permanently associated with them. But they're also resistant, and they've done some work on them as well. And it seems as if the bush pigs have a way of overcoming the humoral immunity. Their lymphocytes seem to survive better. But anyway, they've worked on them as well, and someone had the good idea that you might hybridize them with domestic pigs. Well, they separated evolutionary about 1 million years ago, so the two lines, the warthog bush pig line and the domestic pig line, so you can imagine how eager they are to breed with each other, but they didn't try breeding them, but they tried. They thought that there were pigs that looked a bit like bush pigs, your sort of rather primitive pigs. Every now and then someone said, those have to be hybrids. But unfortunately, now that they do the full genome sequencing on the pigs, they're not. They're just an odd phenotype of a domestic pig. So that one fell through. It couldn't be done. But we did find that there are populations of pigs in the long-time endemic area, our area, which show a much higher resistance. About 40% of the pigs will recover they'll get infected, but they probably won't even show any clinical signs. They just, you'll find antibodies, but you don't know that they've had African swine fever, and they're perfectly safe, and they're not high-grade pigs. But we were thinking that pigs like that might be very useful in areas that don't export their pork, so nobody's going to ask too many questions. And those pigs that have recovered, and they did experiments in Vietnam recently, which showed definitely they don't remain carriers, their offspring are not infected, and it would be a very good way. And what is really interesting is that in countries that have never been infected before, a West African country, in China, they have found breeds that are resistant, seem to be much more resistant. And so everybody's looking very hard at that. They haven't got terribly far with it yet. And another thing that's being looked at, it's actually in Barcelona that they started this type of research of the microbiota of the warthog gut seems to offer protection when it's fed to domestic pigs. And so they're doing a lot of studies on microbiota of resistant pig populations of pigs that show a degree of resistance, like 40 to 50% survival or even higher. And then also on ordinary pigs that don't have any resistance, and on warthogs. And it seems to be quite an interesting line of research to pursue. We've just published a review on it earlier this year, and I think it's nice. I did some work on we imported a small population of pigs from Mozambique that showed this higher resistance But in our laboratory, they didn't, and only one out of about, thank goodness I didn't have to do the infection experiments. I could never have done it, but it had to be done in a high security institute where I wasn't working. So I used to just, sit and get the bad news back home. But it was really unfortunate that it's clearly not genetically directly inherited, that you probably need some sort of epidemiological conditions that would support the resistance.
Yeah, so in other words, there might be multiple reasons why a particular pig might develop resistance against African swine fever, like a warthog or a bush pig, like you described.
Yes, well, one of the things is that we did find, but unfortunately we didn't work on the serology. It's very difficult to work with small pigs, or baby pigs, because they're very sensitive little critters. But the maternally derived antibodies, some of our first lot of pigs, which came from serologically positive parents who'd recovered, their maternal antibodies lasted for 4 to 6 months, which is very long. Most pigs lose them much sooner than that. I mean, most animals lose most diseases. It's lost sooner than that, like classical swine fever. It's gone after 3 months, and it was not in our pigs. And we were also, one of the things we did think about was whether possibly they are exposed when they still have maternal antibodies and may develop a bit of a resistance that way. So there are a lot of things still to look into, but it's rather nice that investigations on innate resistance are now going on all over the place. They've done some wonderful work in Germany at the Friedrich Loeffler on it. They're doing work in China, and we're still looking at it too.
Yep, interesting. What has the main focus of research on African swine fever in North America been?
It's been mainly risk assessment to start with, and it's also been looking at how prepared farmers are, what their biosecurity, whether they're worried, whether they're aware, what they're going to do. And in fact, what we found in South Africa, but it's difficult, most of our commercial farms are either in a particular high health scheme or they are compartmentalized completely. And these compartments are recognized by the government. They're inspected yearly, and they're recognized as free from African swine fever, classical swine fever, which we've only had once in recent decades, and it was eradicated, and foot and mouth disease. But unfortunately, if you have a compartment that's free, or one of these high health farms, and if your biosecurity isn't perfect, or you economize on anything and you cut corners, you get it. So we've had several of them break. And one of them that I looked at recently, they were using a disinfectant that was totally ineffective against African swine fever. And it's actually quite a fragile virus. There are a lot of disinfectants that work for it, but that particular one didn't. And so there's little things like farmers must make sure that the label does say, yes, African swine fever, not just almost any pathogen we will kill, no, that doesn't work. So it has to be really good, and everybody has to be buy-in, and they have to be doing the right things all day, every day.
But that would also mean if you have strict biosecurity protocols together with good disinfectants,
you can stop it.
Yeah.
You can prevent it.
Yep, thank you. That's an interesting insight. Yeah, certainly here in Canada, when you would arrive at the airport, certainly last 2 years or so, there have been these signs about don't bring in food with pork products, etc., just to stop it at the border. Yep.
Absolutely, because that is the other main route of infection, is infected food. And of course, in Africa and in our resource-limited settings, there are a lot of people who feed swill. And feeding swill Well, it's been banned in a lot of countries, but when people are poor and that's the only way they can feed their pigs, you can ban it till you're purple in the face, but you can't have a policeman sitting at every kitchen table. So some very good work was done in Thailand. They're also doing excellent work on African swine fever. They found that they did an experiment. The first time anyone had ever done one, it was all thumbsuck stuff before. It was what you were to do. And people said, well, you must heat it to 70 degrees Celsius for 30 minutes and so on. These farmers that we're talking about, they don't have a meat thermometer, and they certainly cannot use the fuel that they need to boil anything. Or we have to say boil things because that's the only visual measurement of heat. And you can't tell them to boil something for half an hour or an hour, as South Africa tells them. But you can boil it for after 5 minutes. The virus is inactivated. So 5 to 10 minutes of boiling, you're safe. You can feed your swill. So that is something that's been very important, and it's been put into a document on guidelines for African swine fever control in resource limited settings by the FAO that we developed some years ago.
It's time for our famous three.
Every person on your team is wired differently. Some thrive with structure, others drive results through autonomy. Some process information fast and move on. Others need depth before they decide. And when the wrong person is in the wrong seat, not because of skill, but because of how someone is built, performance suffers and no one knows why. Zentrix Insights maps exactly that. Behavioral patterns, cognitive style, and natural work preferences are all visible in 15 minutes. So you stop guessing who should own what and start putting the right people in the right seats. Because the best teams are not just talented, they are well placed. Visit ZentrixInsights.com.
Okay. Do you consider any of the research to have been irrelevant or misleading?
Yes, unfortunately, yes. One of the great things that people have been trying to do is to find new arthropod drivers, because we know that the soft ticks of the genus Ornithosaurus they're the sort of the ones that don't have a hard shield on top, and they're very cryptic. They're endophilic. They live in burrows and they hide in dark places all day and come out and feed for a short time at night. That they were the original host of the African swine fever virus. It evolved in them because it's so unrelated to any of the other animal viruses that they had a look. And then at one stage it was classified as an iridovirus, which is an insect virus. And it's not an iridovirus, but it is closer to them than to others, as well as to pox viruses. The only other arthropod that's been known to be able to, with experiments, been able to maintain sufficient virus to infect a pig for up to 24 to 48 hours is the stable fly, which is a blood-feeding fly. But they've done they haven't done enough work on some of the blood feeders, but they have now been doing midges and all sorts of things, but they've been examining these insects during an outbreak on a pig farm when you would expect a blood-sucking insect to have some African swine fever virus DNA, but it's in such small quantities that it couldn't possibly infect anything else. And I feel that an awful lot of money is being wasted, and it's very unfortunate when they say, so therefore fine screening, mosquito screening all around your pig farm. And that's also asking a lot for people who already have quite narrow margins of profit, because I'm sure your pig farmers complain about that too.
That's a final question I have. If somebody would be interested of our audience in finding out a little bit more about what we discussed relative to African swine fever, is there a particular source or resource that you recommend?
Well, one that I can recommend, if they can get it, it's available as an ebook. It came out last year, is the 12th edition of Diseases of Swine.
Oh, sure, yeah.
You know the book, of course.
Yeah.
There's a chapter on African swine fever that covers a fair amount of this. It gives enough information. I was fortunate enough to be the first author of it, so I found that a very enjoyable experience.
Yep.
And I can recommend that. And then both the Food and Agriculture FAO website and the WOAH have good information as well about the diseases.
Perfect. Thanks very much. So that concludes another episode of the Swine Podcast Show Canada, today with Dr. Mary-Louise Penrith. Thank you very much.
Thank you very much for having me.
You're welcome.